Good morning. So my just title of my presentation is Development of Affordable Biomaterial-based Strategies for Triggering Bone and Cartilage Regeneration. So what are the challenges nowadays faced by these human patients and these animal patients? If we just see from this orthopedic point of view, that the animals and the humans are suffering from these bone disorders like bone fracture, osteoporosis, and also osteomyelitis. And in 2050, it is projected that the cases will be increased to 6.3 million. And apart from these orthopedic problems, sometimes the human surgeon that is faced from this during this fracture repair with the use of these bone plates, and the commonly used bone plates that are used by these stainless steel bone plates, and the second surgery or the revision surgery is needed. And another problem that is faced by these human surgeons, particularly the plastic surgeons and the animal surgeons also, that is there is a chronic non-healing diabetic foot ulcer or wounds, and particularly also sometimes there is a complication leads to infection, sepsis, and the limb loss. And another point, the plastic surgeons, particularly the human plastic surgeon, faces these problems of these deformities, congenital deformities like ear deformities and nose deformities, and that leads to the societal problem, particularly the weaker section of these people, particularly the women for their marital problem. And apart from this, there are certain situations that is during this bone repair process or these orthopedic surgeries that needs the bone substitutes for this repair of this infected bone cyst and the tumor and fracture non-union and delayed union also, and sometimes there is a traumatic or pathological fractures. So presently the existing strategies that are available in this market that are limited, and sometimes there will be some challenges faced by these human and the animal surgeons or the veterinary surgeons, and that has to be addressed by alternative affordable materials that can just overcome this situation. So during that is the excellence of to meet up these challenges. During my more than 2 decades of research work, we have just carried out some research in the silk-based biomaterial in collaboration with Indian Institute of Science, Indian Institute of Technology from Kuwait. We developed some nanofibrous mat from the silk protein fibroin, that is Anthera asama, Bombay exmori, etc., just to develop wound dressing patches, which is affordable and that can help in triggering this wound healing in a very easy way and very comfortable and within a short time. Later on, yes, we have carried out some marine-based material in bone regeneration. Another is this bioceramics, the bioactive glasses, scaffolds, coating on these metallic implants, 3D printed materials for as a orthopedic biomaterial. Another thing is this biodegradable bone plate for fracture fixation and local drug delivery systems for treatment of osteomyelitis. And last one, this animal cartilage, development of this animal cartilage for application of this human plastic surgeries for correction of this rhinoplasty and microcia operation. So these are just from these pictures, because some pictures are here. That is, we have developed some silk fibroin as a wound dressing patches for treatment of this diabetic wound, also from this non-bulky silk fibroin and this bone healing material. And we have seen in an animal model that this type of material is a low-cost material, and that will address for this, that is the curing of this wound, particularly this diabetic wound, and this as a bone grafting material as a affordable cost and very easy way, and very that is a biomimetic and without any toxicity or rejections from this patient point of view. So we have developed some marine-based biomaterials from this coral. That is, we have just converted this marine coral to coralline hydroxyapatite, and we have tried in this animal model so that whether this material is a good osteoconductive or not. We have developed some scaffold from this marine sponge, and we have just observed these bone regeneration capacities. And lastly, that is this from the lobster and these crabs, from the outer cells, we have developed this chitosan, and that is having a good materials for this bone regeneration. So later on, yes, in the human and the animal surgery, we commonly use for these fracture fixation devices are these stainless steel material. But the problem of this stainless steel or this any metallic material is that From there is a leaching of these metallic ions and leads to this metallosis. Just to overcome that situation, we have coated that implants with some ceramic. So just to prevent this leaching, and later on we have trial in this animal and the human model also in collaboration with RGKR Medical College, that we have seen that the leaching phenomenon is very less. And when I was in USA as a adjunct faculty, we have carried out some works on this 3D printed scaffold, and we have also carried out some metallic iron doped scaffold. What is the role of this metallic iron dope normally in human surgery? For this, particularly for this non-union and this delayed union, they use some costly bone-derived growth factors like PMPs, IGFs, and TGF beta, and it has been observed the cost is so high that is one vial of PMPs cost is nearly 40,000 rupees from this Indian currency. But we have just changed this situation, or through research we have seen, if we add some amount of metallic ion, very low cost, that is this strontium, lithium, copper, cobalt, zinc, and if we add with this biac, that is this osteoconductive scaffold, either in this magnesium silicate or magnesium phosphate, the result is at par or is comparable with this that of this growth factors. So we have also carried out with some strontium magnesium silicate also in an animal model. And later on, we have also developed or extracted this hydroxyapatite from this egg cell out of this waste material. So later on, just we have published this work also in common human surgery. There is this bone grafting or the bone fracture devices is one of these fracture devices is this bone plate. And mostly the stainless steel and these titanium plates are used. But the problem of using these bone plates that it needs a second surgery, because after some period of time or the healing is completed, is through a second surgery, we have to remove that implant. So it needs another cost and the patient morbidity, and some patients, they do not go for this revision surgery. So we have developed a degradable implants, bone plates based on from this magnesium alloy, and is coated with some ceramics just to delay the degradation process. And once this bone plates is applied for this fracture as a fracture fixation devices, it need not to be removed in future. So it is an alternative ways, and the research in this globally research is going on. And I hope in the coming days, this type of implant is of immense impact in animal and this human orthopedic surgery. So another point of view, we have carried out research on this osteomyelitis, and nearly 10 to 12 international publications in high impact journal we have published. Osteomyelitis, as you are aware, that it is an infection of this bone condition due to some accidental trauma or with some compound fracture as a sequelae. And the problem is this: when we go for this antibiotic treatment, the amount of antibiotic needed for killing bacteria will not reach to the site of interest because of this blood-borne barrier. That's why we have developed a new strategies for this local bacterial resistance. We have taken some ceramic beads either from this hydroxyapatite, TCP, or this bioactive glass, and we have incorporated some antibiotics within that beads. And these beads are porous in nature, and we have implanted in this bone, and from where it will leach gradually and in a sustained manner so that it will cure this antibiotic.
Sorry for the interruption, sir, but please conclude your presentation in next 2 minutes.
Okay. So another is we have developed some animal cartilage from this animal source and thorough in vitro and in vivo characterization. We have just carried out in animal trial, and lastly for the correction of this rhinoplasty and this microSCA operation in this human patient in collaboration with RGKR Medical College in a DBT sponsored project. So later on, we have just carried out some work on this egg cell membrane, and egg cell membrane as a good materials for this diabetic bone healing. So later, this is a Tata Innovation project from this government of India, that is from the DBT, that is a fish collagen with some bioactive glass doped with copper and cobalt. We have developed electrospinning mats for triggering these wound healing properties in case of these diabetic bones. So this impact and the scalability, we have just undergoing some, we got some patents, and some what is just human application we have done, and large scale application is needed. And in the some stages with the commercialization for this, we have just discussion is going on with some Indian companies for this purpose. So in through which we can just future roadmap, that is development of this biodegradable system, and is a low quality, that is a low cost, affordable, and this that is biodegradable, and that can be used for this as a tissue engineering devices we have done. So these are these I acknowledge with my honorable Vice Chancellor, CGCRI, the Department of Biotechnology, ICR, IIT Kharagpur, Guwahati. And during my 2 decades, I received 5 national awards. There is John Nehru Award, National Bioscience Award, Biotech Product and Process Development and Commercialization Award from this honorable President of India, CSIR Technology Award as a award-winning team, and I become this fellow of this Indian Society of Veterinary Surgery, National Academy of Agricultural Sciences, and in the last month I have been selected as a fellow of this Royal Society of Chemistry as a maybe as a first veterinarian in this country. Thank you all.
Thank you, Dr. Nandi.
Yes, sir.
It's quite interesting. But tell me whether it is still a research stage, or have you got approvals, and if they are approvals from appropriate authorities, how many you have got?
So regarding these approvals, that is this from this animal trial point of view, we have got this approval from this institutional animal ethical committee, and from this some work we have carried out in human experiment, that is from this as a part of this project from this DBT sponsored project, we got this approval from this ethical committee of this respective institute from this medical college. And we have applied for this DCGI for fruitful application in a large scale basis.
And how many are in pipeline?
So pipelines are 3 to 4 projects are in pipeline stage. So we have to, after getting this approval from this DCGI, we will carry out for in a large scale for this application in this human basis.
So which one you are finding most effective, whether silk based or marine based this thing, and are they, do you think they are equally effective on humans as well, or they should be?
So the first thing, these all are equally important, but I think this biodegradable bone plating system that is very much important, because if you just see these orthopedic problems in the nowadays, this very number of patients are admitting for correction of this, that is the fracture devices. So this area, that is the biodegradable fracture fixation devices using magnesium-based alloys, and they are coating, that is the most interesting part from my research work.
Just last question. Are you collaborating with the, allopathic research also?
Oh yeah, actually.
Yeah, because you have mostly done on animal, from animals point of view, no?
Okay, actually I have some good collaboration with RGKR Medical College for the plastic surgery department. We have already carried out in collaboration 2 projects from this DBT sponsors project with cartilage grafting, animal cartilage grafting for these human patients. And another project we have already completed from this ACRB, that is Science and Engineering Research Board from this TST, that is a treatment of osteomyelitis through local drug delivery system in collaboration with RGKR and the Central Glass and Ceramic Research Institute.
Good. See, most of the research remains in lab, so we keep on emphasizing lab to land. So it must take shape. Ultimately, it must benefit the animal kingdom or the human.
Sir, as you know, just for that is for commercialization and for the human application, so we have applied for this DCGI approval, and after getting it, we will go for this different. We will approach for these different hospitals for large scale applications.
So no patent you have received as of now.
Already from this, we have developed one patent from this marine mollusca that is granted in 2018 as a novel anti-cancer agent that is granted.
Okay, all the best to you. Thank you.
Thank you, sir.
Dr. Samir,
Yes.
You have developed some materials for transplanting into humans and animals for their to increase their bone life.
Yeah.
Have you, during your research, have you also come across any idea that if some supplements can be given to human or animals, which increase their bone density instead of transplanting at a later date.
Okay, actually, regarding this bones and density, the common problem faced by these humans, that is this osteoporosis. And this osteoporosis is common particularly in the women after this post-menopausal stage. So in animals, at least during this as an experiment, we have trial for this implant, and as it is during that we have not given a such supplement, but still the normal food along with some allopathic medicines as a supplements we have just added. And now at these moments we are working on this gene nanoparticles for triggering this wounding process. We are searching for this oral medication. One part we have just kept for this oral medications as a oral therapy, and apart from this application as a disinfect.
Because generally doctors prescribe only calcium and vitamin D. For
Yeah.
osteoporosis. There is a need for some other nutrition particles which can increase bone density and longevity of their bones.
Actually, in human, if you just see that just zoledronic acid is there, and bisphosphonate there is there. That is the compound they are using for treatment of this osteoporosis. But the problem is this: it is a costly one, and many of these particularly the poorer section of the people, they cannot afford for this costly treatment. That is the main that is this hurdle for that one. Still, they are using these bisphosphonate and these zoledronic acids as a supplement for increasing this bone density. Apart from use of this nanocalcium, that is the calcium as a silcal or this type of product, and sometimes some vitamins, particularly which will increase their bone density.
Thank you, Dr. Nandi. Very good project.
Okay, thank you, sir.